All right, I'm coming to get your test. If there was a question, you can let me know. But right now, you are filling out these notes. And can somebody go get Ian?
I want to stay in here for the guarantee. Yeah, but
See the big skull and crossbones on it? Did I get your test?
Yeah. I have a C in history. I'm test. I couldn't do test correction. No, because at the end of try. These triad systems so stupid. I tried doing this so far. It's literally right? And they decided to give you all the work at the last week.
All right. So you guys started talking about stem cells yesterday and differentiation. So now that we have an idea of what these eukaryotic cells, we're mostly going to focus on eukaryotic cells in this unit. We know that there's also many different types of cells, right? We have these cells that are specialized. So this whole unit is how did they get there? How did they get specialized? And it's through this process of differentiation. You think about when you were 1 day old, you were one cell baby, right? And that one cell was just one cell. And then through your development over the 9 months, you went from one cell to tons of cells, and each of those cells has a different function, right? Your organs developed, your bones developed, your brain developed. Yes. We need to go back one. Okay. Page 4. Go ahead. So through that development process, what was happening was that one cell was dividing and differentiating, specializing into many different types of cells. It's a pretty fascinating process if you look at the development of an embryo into a zygote, a zygote into an embryo, into a fetus. Lots of crazy things happen. And then by the time you are born, you are pretty much a fully formed human. Still a couple tweaks to be made. Like when you're born, your digestive system doesn't fully develop until about 2 years old. Your bones, as you guys know, babies have soft spots in their head, right? That's because the bones need to still harden and come together. But for the most part, by the time you're born, all your organs and everything is all pretty much intact. And that's thanks to differentiation. Okay. So differentiation is regulated by certain proteins that bind to specific base sequences in DNA. So basically with your DNA, there's different ways that it can be expressed, kind of going back to this slide. We know that we have DNA, which is split into genes, and each of those genes goes through transcription, translation, makes proteins. Okay, and those proteins and other components come together to make tissues and organ systems. But how that transcription and translation happens is regulated by these proteins. Okay, there's certain proteins that can turn things on and turn things off and edit how things develop. Okay, examples. So this is connected. Yes. What do I want you to write for example? Other examples, you have bullet point examples. The making of egg and sperm both involve mitosis, cell growth, meiosis, and differentiation. You could just say the making of egg and sperm. You can leave it there. The making of egg and sperm. So if you look at this image, we have something called a gonocyte. This is a very primitive cell in the development of yourself. That goes through differentiation, means it's going to specialize a bit, and it will turn into a spermatogonium. That spermatogonium will go through mitosis and meiosis, and a little bit more differentiation at the end to eventually become a sperm. Okay. Another example here, we have, actually this picture was at the bottom of page 4. But a lot of differentiation occurs during embryonic development. So in terms of when differentiation happens, most of your differentiation is happening before you are even born, okay? So in the beginning, as your zygote is developing, turning into an embryo, that's basically where most of the differentiation happens. There's not a whole lot of differentiation happening at this point in your body. You're all pretty much already specialized. So for the second example, you could just put during embryonic development, knowing that it mostly happens in the beginning of life, the beginning of the development of the organism. Now, this is science. Well, it does make sense that you would specialize very early on. Yes. That's good. All right, this one, we should get some highlighters. If you don't have a highlighter, you can grab a box. So something that can affect or alter the differentiation is sometimes the environment. So this is kind of a cool example. With certain animals, there is a specific protein responsible for coloration of skin, hair, fur of many animals. And this protein is called melanin. Right? If you are a darker colored human being, you have more melanin in your skin. This is true for animals as well. This melanin, this protein, is regulated by a certain enzyme called tyrosinase. Okay, so that's true for a lot of organisms, including humans. But these two specific organisms, Himalayan rabbits and Siamese cats, have a special gene. And this gene is a temperature sensitive tyrosinase gene. So this leads to the environment having an impact on what they look like. So if it is cold, they will have darker fur. The cooler the body, the darker the fur. No, you don't need to color the bunny. So the genes are expressed differently depending on temperature. So a very hot topic, I think this was said in a previous slide, of genetics is looking into this gene expression. We have now sequenced the genome. We know what genes do what for the most part. But how these different things get expressed is still kind of a mystery in a lot of cases. But we know for this example that the environment, the temperature affects how much melanin is produced in the organism. So it's kind of a interesting example. That's not for humans. That's only for the rabbits and the Siamese cats. Okay. Next, we have flower development here. The development of a flower by an angiospermatophyta is due to differentiation by cells in the shoot apex. I don't know how much you know about botany, but basically you just want to understand that the change in the gene expression is in response to the change of the light-dark cycle in the environment. So again, environment is having an effect on gene expression. So I did want you to kind of color code this. So in your notes, you have the different boxes. Basically, you want to color dark green box and then dark green area. Real quick. It doesn't have to be perfect. Should get dark green, either yellow or orange, light green, red, blue. It's what? So what you're seeing, I wanted to give you a second to get your colors out, but what you're seeing is each gene is a different color. So as you can see, these three different pictures, different genes are being expressed in different locations and different amounts. So in the first picture, you only have the first three genes that are being expressed. Okay, next, your light green pretty much goes away, so that's being expressed a lot less. And we have added the SPL4 gene and the SPL5 gene. So basically in the second picture, you're seeing that the start of a flower is happening. In the first picture, it's just green matter, for example. And then in the second picture, you'll see that this is where you'll actually start to get a flower bud forming. And like we said in our highlighting, the timing of this, when these genes are being expressed, is affected by the environment. In this case, the light dark cycle. Some plants will only start to bloom flowers at night. Some plants are during the day, depends on the plant. So when you think about, like, let's fast forward to you studying for the test. This is not something I'm going to test you. I'm not going to be like, in which picture are you going to have SPL4, right? From this picture, you want to see that you're having a difference in expression of genes at different times. Okay, we don't always want to express every single gene in our body. Does that make sense? For example, maybe antibodies. We don't want to spend energy constantly producing an antibody to fight a sickness that we're not currently feeling. So we only want to express antibodies when we have that in our body, or generally. All right, we're moving on in 10 seconds. Five seconds. Remember, these are posted if you need to go back and look at them. Okay. So in terms of stem cells, one reason you may have heard of them is because they can be very useful for research purposes and also for therapeutic uses. Something that we recently voted on, last major voting time, was whether we should spend money on stem cell research. So as an adult, you had a choice. Do I want to vote for stem cell research or against? Specifically, the money spending part. okay? Because you could be for stem cell research, but maybe you don't want your taxes to go towards that. But that was something we recently in California voted on. So the capacity of stem cells to divide and differentiate is very interesting to us humans. If you remember the video yesterday, certain organisms can completely regrow like a half like of a body, right? We can't do that. But would it be cool if we could? And how we might get there is by studying these stem cells. Yes. Yes, and something we'll go into later in this unit is where we get these stem cells from, because along with this ability for us to study them and research them and the possible therapeutic uses comes ethical debates. Well, that's your opinion. There are people with opposite opinions. Actually, I don't remember. I should look it up. I thought I had it in this presentation, and actually maybe I do, and I just forgot. But I'll look it up. I can't remember. Okay. Can somebody turn off the lights, please?
What are the odds that they are listening to the exact same playlist? Probably pretty low. After all, everyone has very different tastes
in music. Now, what are the odds that your body will need the exact same medical care and treatment as another person's body? Even lower. As we go through our lives, each of us will have very different needs for our own health care. Scientists and doctors are constantly researching ways to make medicine more personalized. One way they are doing this is by researching stem cells. Stem cells are cells that are undifferentiated, meaning they do not have a specific job or function. While skin cells protect your body, muscle cells contract, and nerve cells send signals, stem cells do not have any specific structures or functions. Stem cells do have the potential to become all other kinds of cells in your body. Your body uses stem cells to replace worn-out cells when they die. For example, you completely replace the lining of your intestines every 4 days. Stem cells beneath the lining of your intestines replace these cells as they wear out. Scientists hope that stem cells could be used to create a very special kind of personalized medicine in which we could replace your own body parts with, well, your own body parts. Stem cell researchers are working hard to find ways in which to use stem cells to create new tissue to replace the parts of organs that are damaged by injury or disease. Using stem cells to replace damaged bodily tissue is called regenerative medicine. For example, scientists currently use stem cells to treat patients with blood diseases such as leukemia. Leukemia is a form of cancer that affects your bone marrow. Bone marrow is the spongy tissue inside your bones where your blood cells are created. In leukemia, some of the cells inside your bone marrow grow uncontrollably, crowding out the healthy stem cells that form your blood cells. Some leukemia patients can receive a stem cell transplant. These new stem cells will create the blood cells needed by the patient's body. There are actually multiple kinds of stem cells that scientists can use for medical treatments and research. Adult stem cells, or tissue-specific stem cells, are found in small numbers in most of your body's tissues. Tissue-specific stem cells replace the existing cells in your organs as they wear out and die. Embryonic stem cells are created from leftover embryos that are willingly donated by patients from fertility clinics. Unlike tissue-specific stem cells, embryonic stem cells are pluripotent. This means that they can be grown into any kind of tissue in the body. A third kind of stem cells is called induced pluripotent stem cells. These are regular skin, fat, liver, or other cells that scientists have changed to behave like embryonic stem cells. Like embryonic stem cells, they too can become any kind of cell in the body. While scientists and doctors hope to use all of these kinds of stem cells to create new tissue to heal your body, they can also use stem cells to help understand how the body works. Scientists can watch stem cells develop into tissue to understand the mechanisms that the body uses to create new tissue in a controlled and regulated way. Scientists hope that with more research, they can not only develop specialized medicine that is specific to your body, but also better understand how your body functions, both when it's healthy and when it's not.
Okay. So that basically went over kind of a lot of the things that we're going to go over next in our notes. While you write this down in your notes, the bill that I was talking about says, this article was from November of 2020, which is when the voting happened, right? Voters in California extend life of stem cell funding agency. So it was called Proposition 14, passed with 51% of votes. So it's kind of like a barely pass, right? 51% of votes. Authorizing the state to issue $5.5 billion in bonds to give the California Institute for Regenerative Medicine a second lease on life. So basically, the California Institute for Regenerative Medicine was running out of money. So they proposed to California, please give us more money so we can keep doing our research. And California, 51% of us said, yes, have $5 billion. This is coming from bonds. So this is where my knowledge gets a little limited, but I think with bonds, it's partially coming from people who choose to donate money to the cause, but it could also be coming from some of our taxes. That I'd have to look into. The agency announced last year it was running out of its first $3 billion, which was approved by voters in 2004. With the new bonds, the agency will now continue to fill what supporters say is a critical funding gap in stem cell research and fund ongoing clinical trials and training programs for early career researchers. Of the new funding, 1.5 billion is to go toward projects developing therapies for neurological disease, such as Alzheimer's, Parkinson's, and dementia. This guy says, we are thrilled. Of course, $5 billion, I'd be thrilled too. Despite a $20 million strong campaign in support of the measure and the absence of organized opposition, Prop 14 passed by just 325,000 votes out of more than 15 million cast. Here we go. It will ultimately cost California taxpayers $260 million a year for 3 decades to pay off the bonds. So we're going to work together for the next 3 decades to pay off this money. Taxes are going up for lots of reasons. That's one of them. Okay. I need to get you guys some homework. I know. All right, I need you guys to do slides 40. I know. To 40 to 48. Can we do 8 slides? I think we can do 8 slides for homework. One of them's a video. 40 to 48. We get 32 to 39 for class. Okay, you guys might want to write that down somewhere. So that gets you through page 6 and 7. So you're going to do page 6 and 7 using slides 40 to 48. Go ahead. Well, class is over. Might as well just wait. Yeah. This, yes, and the presentation. Yes. You're in the Google Classroom, right? You're writing for homework. You're doing pages 6 and 7 from slides 40 to 48. I'll also send a reminder. So you should add a reminder. Okay. Monday. Here's the remind code. All right, you guys. As you know, we have assembly schedule. I have the schedule written on the whiteboard if you want a picture of it, or oh, did you? All right, bye guys. I'll see you tomorrow. I'm just writing this down for you. And we're going to like 5, 6. Okay. Sometimes it's before, sometimes we have to be here. I'll be here. Yes. Did you have a question on the test too? Yeah, you still got, okay, you still got 6 points out of 8. I think I needed more on what the Golgi does, prepares, but how does it prepare? Also, you didn't have the information about starting from how we get the instructions from the nucleus, and the instructions go to the rough ER. Yeah, so there's a few more things you could write, but you still got 6 out of 8. Okay.
I ran out of time and I wrote naked right in my DNA. Oh my god. I think that's why I got one taken off on the like what's the difference because I wasn't that
Yeah.
sort of thing.
Wait.
But you got one more than me. So on the lowest of our table. Wow.
Right.
Ready to go.
So, we're not filming today, so I would like to film with you.
Oh, they're kidding. You still need us there?
Not necessarily, I mean, here's what we got, we finished the first thing.
Oh, yeah.
So now, this is our plan, I think that we've got, we have everything that we need, we're gonna edit it together, down to Mr. Lynn, next week, show it to him, and tell them what supplies we still need, tell them what we need to fix out. So it's like our trial one, but I'm pretty happy with it.